All right. Hello, everyone. My name is Bogumil, and I'm leading PharosBio. And I have a couple of videos in there, so if something doesn't work, don't kill me. That's an experiment of making it a little bit more inspiring and engaging. All right. So a light bulb is a symbol of human ingenuity. And we invented it 100 years ago, and we didn't stop innovating. So we've built cars, we've built planes, and we also conquered the moon. And this constant urge to exchange ideas, innovate, made us to build a computer and the internet. And in biotech, we also were not left behind. 50 years since the discovery of double helix, we sequenced the complete human genome and also learned how to modify it using CRISPR-Cas proteins. However, one challenge still remains, where we are really slow and inefficient. Drug discovery cycles take more than 10 years, and 90% of drugs never reach the market. It's a big issue, and it highlights a couple of problems, mainly slow R&D cycles. And partially they can be attributed to long wet lab validation, but the larger issue at hand is how we come up with ideas, experiment, and strategies. These days, we face a new tech revolution. AI went from beating Garry Kasparov in chess to solving protein folding challenge using AlphaFold. And this can partially be explained through increase in computational power, but also increase in dataset sizes. Biological data is vast, and scientists these days analyze it using manual tools. We surveyed dozens of researchers, and they mostly reported Excel, Prism, Benchling, Python, or R. Unfortunately, there's just too much information out there to analyze it manually. One example of this is when you look at the number of publications released every year, there's over 3 million. Let's assume 1 million is relevant for a given researcher. If a researcher can read 3 papers per day, he will access 0.1% of the available knowledge. So what it means is that 99% of ideas that the researcher could use will never be reached. And this is only one of the examples of how vast biological information space is. So how do we tackle this problem? Well, we can combine existing knowledge bases that we generated over decades of research with autonomous AI agents that can use biology-specific tools. But so, what are AI agents? How many of you heard this term and feel like you understand what it is? Show of hands. 1, 2, 3, 4. Okay, cool. That's great that I made 3 slides for it. So let's start with a general large language model. You can pose it a question, and if the answer is in the training dataset, then it will provide you an answer. But if it's not there, then you will get some weird hallucination. So we can solve it using compound systems. One example of this is a scientist could ask which mutations of protein X improve binding to the protein Y. And if you ask this question to a general LLM, it will most likely fail. But instead, we can take this query and convert it into a search query against ScampI database. And if both proteins are there, most likely you will get a correct answer. So we can see this shift from model-centric approach to compound system-centric approach. And this is because these days the large language models, they have billions of parameters. And even if you retrain them every month, in this month you will have a multitude of information that you will never have in the model. While compound systems, they are really modular and easier to adapt. But then you might ask, okay, so where is the agent in it? The agent comes with control logic. So normally, we control the flow of the workflow using programmatic approach. So we can make a stringent workflow with if-else statements, and it will decide how the software will analyze the data. And this is very stringent, and it will work if the workflow has to be really robust. But these days, you can put an LLM in the steering of how the workflow will work. So the LLM can decide where to take the flow of the program. And we can consider this as a slow thinking system because maybe the agent will iterate many times before it reaches the answer, but it's also very flexible. So what properties does the agent has to have? One of them is to reason. What does it mean? It has to be able to plan its course of actions. Another one is to act. So the agent has to be able to use tools. A tool in the industry term would be search for a database or running a Python script or R script or using a tool like AlphaFold or Autodoc. And also an agent has to have access to memory. And why is it important? Well, if in the first run an agent writes a script that throws an error, then in the second loop it can correct this error. Okay. So how does it work within the context of biology? I will show you one example that we tried. Imagine you run a large hit screening. You found the right hit, but you don't know yet how it works. So you design CRISPR knockdown experiment and you have a dataset for it. You can upload this dataset and pose the question, find potential drug targets. An agent will prepare a plan for it, write a Python script that will probably look through your list of hits and fold changes, that will sort them and execute the script and come up with top genes, the genes with the highest fold changes, for example. And this is just first iteration of an agent. What would happen next is probably taking this gene, searching for a database of publications, looking up how this gene could be involved in the disease mechanism and so on. And then the agent can report it. So I will show you how we did it within the platform. The way it works is that scientists can connect all of their databases or upload the spreadsheets directly. In this case, we upload this CRISPR gene knockdown dataset. This is saved and we can start a new conversation. That's how the dataset looks like. And the scientists can pose a very general question like find potential drug targets and set it to auto mode. And in auto mode, the agent will choose the right questions to ask, write a code to analyze the data, summarize it with summary statistic plots, and interpretations. And the interpretation is based both on the table and the vision model that looks at the plot. And what is nice is that the agent also searches through the papers. So when it interprets the data, it does it using the literature. And at the end, when a scientist is satisfied, he can take all of those results and summarize them in a report. And this is a fully AI-generated report. And in this report, we can see all of the results, so plots, summary statistics, tables, all of the conclusions, and also references to the literature. And the fun part about this report is that we actually found the same gene as the researchers that provided us with this dataset. So we basically recreated part of their research paper in the platform within a couple of minutes. And let me tell you a bit about other features that we're working on. One of them is deeper research. And the whole idea here is that maybe when you use a vector database, you will retrieve first 5 publications that are not so relevant. But based on those publications, normally scientists would go back, learn a bit more about the topic, and continue digging until he has the answers. And that's basically how the deep research works. So we pose an initial question, we search through the database of publications that we vectorized, and then we basically stimulate the agent to decide, do we have enough information? Should we continue searching? And once this is complete, we can either have it as a deep report, or we can do something else with it. We can, for example, use it to extract numerical variables from the publications. So we did an experiment last week with this, where we took papers that are focused on IPD, and we had several variables that we wanted to extract. It's clinical data where we want to see whether patients improve in the 6-minute walking test over time. So we've built a workflow where the agent can take all of those papers, find the right clinical results, extract them as per study extraction, and also using our code execution tool, build pseudo data set of these clinical results and report it. And the whole end-to-end workflow took 15 minutes, and we reached 86.67% accuracy. So maybe now you start seeing what we're trying to do here. If we can combine this deep research with extracting the variables and automatic reporting, we can basically do a meta-analysis, right? So we can extract the right variables from the right papers and automatically summarize them. So this is something we tried recently. For now, we're slightly below 90% accuracy with 1 week of development. So I'm pretty hopeful that it can get even better than that. And there are very many similarities within, how the tools are used within the large language model space. So some of the ideas of what are the differences between Pharos approach and typical ChatGPT approaches. I think especially for organizations, what's important is that if you have proprietary data, we can include it in our knowledge base and we can dedicate a whole server for only your data so we can keep it secure. We can also use local models. So if an organization is scared of data leakage, maybe there is something very sensitive there, we're free to use local models and we're really building around science. So we already vectorized all the publications in cancer research until 1950s and everything else 5 years backwards. And we're building with biology-specific tools. So this is something that OpenAI maybe wouldn't really think about or wouldn't really find value in. But if a researcher has a tool that they really need to use, we could include it in the platform and let agent use it. And yeah, this is just some of the ideas of what is a wrapper, right? It's a really common term in large language model space. Everything we're building these days is called a wrapper, but there's a lot of development that has to be built around the models in order to make them work. And one example of what people these days don't consider a wrapper, but it's a very common tool, is, for example, Netflix. Netflix is completely built on top of Amazon servers. So we could consider Netflix as being a wrapper for Amazon. And slow wrap up. So we have, we're still early stage, but we have great collaborators. We've completed pilot project at one of the BIA companies, Dandelion, deployed for all employees at Cirqle. And we have strong academic partners, including one letter of intent from Boston Children's Hospital. And how do we differentiate from different tools? As I mentioned at the beginning, majority of tools within science are very manual, while we want to go as autonomous as possible. Of course, these days the models and technology is not there yet, so I would consider it semi-autonomous with human intervention. But that's where we position ourselves. And we have a great team. So I started the company with Aurel. We both have more than 5 years of experience. Me more on the industrial side in biotech companies, while Aurel through his academic career and his postdoc at Danish Cancer Institute. And also we have great developers from top research institutes like Karolinska and University of Cambridge. And we're supported by experts from business and research from top universities like MIT and Harvard. And just to give you an idea, right? So for now, we have a data analytics platform. We're making data analysis faster. We can search through millions of publications that would not be accessible to human in his lifetime. But the idea is greater. So we can see this progression of technology from Garry Kasparov being beat in chess to AlphaFold. And the same progression can be seen within the automation space. So there's much more automation within the laboratories that's happening. There are several cloud labs already. So we hope that with this increase in automation, both on the software side and the hardware side, we can combine both those tools. And the model will not only be able to come up with ideas, but also validation strategies that can be then translated to scripts for, let's say, liquid handlers in the lab to really close the loop of science and deliver life-saving drugs on demand. All right, so thank you for your attention. If you would like to try the platform, then feel free to use the QR code. And we're running a small pilot with a limited number of researchers. And we would be really happy to get your feedback. And if you would like to work with us, then we also look for business and software developers. So yeah, also feel free to scan the QR code. It will get you to our website, to the contact form. But also feel free to approach me afterwards and we can chat about it. So thank you. That's all from me.
Excellent talk. Thank you so much. Let's see if it sparks some questions in the room.
Thank you for a really great talk. That was really interesting. So I was a bit unsure. You gave this example where you said you had 86% accuracy.
Yeah.
Did you give it 5 papers as input? So,
yeah, because it's a trial we're doing with one of the partners, but I thought I will give you a kind of glimpse to it. So we knew that those papers will contain the information we want. But the idea is that maybe with combination of the deep researcher, we could find the right papers, right? So you need to optimize a couple of steps there.
Yeah.
There is a step of finding the right papers, extracting the right variables, and then writing the correct code to analyze them. So this 86 is compared to the ground truth that we manually extracted from the papers. So it's extraction accuracy.
Okay. Yeah, because I was just, so it sounds like you're not really there yet, but what would happen if you were giving it like the full Google Scholar?
Yeah, we will see. Maybe that's going to be next talk.
Yeah. Cool. Thank you.
Yeah.
Yes, we'll definitely invite you back to give a talk when that happens. Any other questions in the room? Yeah.
We have some online. Now, I'm not so much within it, so I try to read out the question as good as I can. So it's from Nikolai Lulasen, and he's asking, in your experience, how hard is it to get LLMs to parse tables and figures out of the box? Do you use any special tracks for OCR on tables, figures?
Yeah, so it's a good question. Basically, with those PDFs, we extracted text data from the PDFs, so there was no OCR necessary. We tried with OpenAI vision model to extract also visually, and it sucked. Yes, that's as much as I can tell now. I think there is a value in this extraction, OCR extraction from the paper documents, but because still many companies use paper. But we didn't try it, so I can't tell you much. But it's pretty good to extract text from text. Text from image, we didn't have good experience.
Thank you.
So how do you see a tool like Site.ai? I didn't see that in your landscape, but they also extract information from scientific literature.
How are they called? Could you repeat?
SCITE.ai.
Yeah, I don't know those guys.
Okay.
But I assume I can make an educated guess that they focus really on extraction, and for us it's building agents. So we want agents to use tools, and extraction could be one of the tools that they will have in their repertoire, right?
Do we have more questions? We do. Did you have one?
Yeah, quick question. So how does your system decide on which hypotheses should be tested? Because a data set could have many different potential hypotheses proposed to be testable by a data set. So
yeah, so that depends on how deep down you want to be. So if you have a really specific question you want to ask, then we can provide an agent with your specific question, and then it will run once and it will try to answer it. But if it's a complex task, it will try many questions until it reaches the right solution. But we don't have prioritization of hypotheses yet. So it's one of the ideas and one of the problems that many researchers told us about. With hypothesis is one, right? But it could be also with you would like to maybe prioritize protein mutants, right? You have a list of maybe many proteins that you optimize, and which one should we pursue next, right? So we haven't looked into it that much yet. For now, the agent gets kind of the free realm. So if we leave it for a number of iterations, it will come up with questions, and by the end summarize them and provide you an answer. But whether it prioritizes the right hypothesis, we still don't know. It's something to look through. It's a good idea.
Yeah, really good question.
Yeah.
Hello. So Daniel from Novo Nordisk, and I sit in drug development also. So very interesting speech. So thanks for that.
Yeah.
So one thing is around the 10 years. I think we can debate whether it still takes 10 years, but that's one thing. So again, perhaps a little bit on your commercial model. So you primarily look at the research part here and getting the molecules out there. And just a consideration around what's your commercial space, because I think also to the gentleman over there mentioning the saturation in the market, there are many people looking into exactly the space you're in. So just what's your considerations? What's your commercial model to the companies? Are you looking for mid-size or big pharma? And your also, perhaps, I know this is getting a lot of questions, but the last part is around the authorities, because one of the big things in the 10 years or 6 or whatever we are looking at is that when you then talk to the authorities and start to talk about AI, you get into some kind of a squeeze there. So some considerations there would also be nice to have. And sorry for the many questions.
No, very good. I think it's fair to say that we're still figuring out the best sales channel. The whole idea with Pharos started from a very industrial problem. It was not so research-oriented. So the problem was really at the beginning that there is so many databases within big companies. At Novo, you probably faced it pretty clearly. And the Pharos was initially built to build you connectors to all your systems and then ask questions across all of them, right? So that's like a very, very industrial challenge to solve. But at the end, we want researchers to use it, right? So if we connect the systems, who will at the end analyze this data? Researchers. So that's why we also want to make sure that the analysis part is valuable to the hands-on scientists. So from the perspective, let's say, of a project manager or R&D manager, he can see, all of the questions that researcher asks, all of the analysis and the reports that are fully reproducible. But if the scientists are not satisfied with the answer, then what's the point, right? So it's kind of combining both. The management has the overview of what's happening within the specific team, and the researchers have faster and better analysis.
Thank you very much for a very interesting talk and presentation. My question is still on the quality of the input that you are getting and taking into consideration the meta-analysis as an example. Yeah. How do you, can you give a little bit more detail in terms of how do you intelligently search? For example, there is a difference between an open label study and a randomized clinical trial. And then how do you assess the methodology was done right, the sample size, and so forth?
So we don't evaluate whether the study was right itself, right? It all depends on the questions. So like the way we did it was that we left LLM with 4 text blocks extracted from those papers, right? And then we provided with a schema of what variables do we want to extract. So you could imagine like one variable was, for example, mean at baseline for a control, right? And you could imagine a schema as a table where there's one variable, and then you have to, like, as a scientist, you would explain what exactly do you mean by that, right? And then the LLM will try to interpret what you mean, so the context, and will look at the paper and extract the value the model thinks is the best, right? But so basically, it's hard to tell whether the model thinks about it correctly. The only way to tell is by comparing it to the ground truth. So that's what we did, right? But it's really based on how well you define the problem as a scientist, right? So for example, we had this discussion with one of our partners about the dropouts in the studies. So we started with, let's say, 10 patients, and then we were left with 8 until the end or something. So how do we calculate? Do we include all 10, even though the measurements are missing or not, right? So this is something a human would have to be really explicit about to extract the right value. Otherwise, the model will select, either of them. Either it will assume we take 10, or it will assume we take 8. So there is still this human and AI interaction where you have to be really precise. The more precise you are, the better variables you get at the end.
Thank you.
And also, just to say, like we just finished this, so it's a week of development. So it's gonna, we will get more data, and then most likely it will be also clearer how to tune those models to get better answers, right?
So we have time for another question if somebody has a burning question here. Yeah.
Hi, thank you. I actually have a startup question. First is, how do you plan to monetize this product? And second is that you mentioned that you got 3 letters of intents. And could you maybe talk a little bit about how you approach and how did you end up with letter of intent?
Yeah, we started as a spin-out from another company, and we already were kind of close to the BII community. So that's where 2 out of 3 letters came from. And then remaining one was through my co-founder, who was a little bit better connected with the Boston's Children's Hospital. And that's how we could present the solution and get the letters of intent. So you know, connections, personal connections, right? That's where we started. Yeah.
How about the monetization model?
Yeah, so we did kind of a short sum up on, you know, how big is the opportunity there. And I estimated that there's around 4.5 million researchers out there. And when you look at the solutions that scientists use in many of the big pharma, not Novo yet, I think, a lot of them uses BenchLink. And BenchLink, at least when I was buying it for the company I was working with before, costed $500 per user per month. So that's how we imagine the pricing for the solution for the enterprise partners, right? And that leaves you with a $27 billion market in the B2B SaaS space.
Yeah, thank you.
That's all we're going to have time for this time around. Let's give Bogumil a big hand of applause.
Thank you.
Thank you.